Good day. These are dihybrid cross notes. Most of the time you're going to do monohybrid crosses, and that means you're only going to look at one trait at a time, and it's pretty straightforward. So on the test, when we get there will be a section of questions about dihybrid crosses, and it will tell you that you're dealing with two traits at one time. Di means two, and so we're talking about two traits that sort independently. It has to do with the law of independent assortment, and we are looking at what are the possible gene pairings when we get two traits at a time. So we're going to look at rabbits here, and so we could have dominant trait is spotted, recessive is solid for pattern, and for color dominant is black and recessive is brown. So it would be a capital S for spotted, little s for the solid, capital B for black, little b for brown. So we're told we have brown spotted and black spotted. And we say, well, what's going on here? Well, brown, recessive, so we have two little b's. And then we are told it happens to be spotted, and we're told it happens to be capital S, little s. This has to be given to us. If it's spotted, we don't know if it's homozygous or heterozygous. So I'm telling you it's heterozygous. Same thing here, I'm telling you the black is heterozygous and that the spotted is homozygous. Well, probably the hardest thing is figuring out all the possible combinations of genes to load in here, essentially for both parents. So these would be the genes in the egg and the sperm that are going to unite, and these are the possible crossings we could get from organisms or rabbits that have these genes. So we go little b, big S, we get that, and then this little b can get together with the big S. This little b can get together with a little s, that's why I write it here, and this little b here can get together with that little s. So we're pairing the first letter with the last two letters, and then the second letter separately with the last two letters. We do the same thing here. We get big B, big S, and we get big B, big S. Then we get little b, big S, and little b, big S. So I hope that makes sense. But once you've got that done now, it becomes really straightforward. Because you look above and left, and you go big B, little b, big S, big S. Big B, little b, big S, little b, big S, big S, little b, big S, big S. And then we do the same thing next row. And it turns out to be identical because it's identical crossings. And then this one will be different because we have a little s here to combine with a big S. So it will do this. Little b and a big B. We always put the capital letter first if we have a capital and small letter that are same letter. We get those combinations, and then this was same crossing. And so these two rows will be identical, last two rows as well. Now we go back and we have to interpret these things. Well, we say big B, little b is black, and big S is spotted. And this is identical here, and little b, we know that's brown, and we also know big S is spotted. And so all we do is we interpret all 16 boxes and then establish some ratios. And what we get are 8 black spotted, which is 50%, 8 of 16. 8 brown spotted, 50% chance of those. 0% chance of black solid, and 0% chance of brown solid. So that one is done. Now we carry on to another example. Here we have big B and big S, big B and little s, little b and big S, little b and little s, and I filled those in here. Then I look over here and I see it's identical to what I had on the left, so it's going to be identical. Those listed down, these will list across, and this one is going to be a little more interesting as we go. The filling in is very easy. Look for the B's above the rectangle, look at the S's above that are next to it and above. So there's a B, big B, and a big S and a little s there. And here we have a big B and a little b. And there we have two big S's. And then we have big B, little b, and then we have big S and little s. And we just carry on like that for each row. So make sure you get this copied out. Make sure you understand how it's working. If I look at any of these rectangles, I've looked above and to the left for the letters coming together, the B's and the S's. Then I have to interpret it, and this one's going to have quite a wide range. So you may even want to do like a tally chart. So you might want to say how many are black spotted, how many brown spotted, how many black solid, how many brown solid. And typically it's fine if you just go with the ratios. These are all out of 16, 9 out of 16, 3 out of 16, 3 out of 16, and 1 out of 16. There's only a 1 in 16 chance of getting all recessive genes combining in this case. And here I've just written them as percents. So that's how you deal with it. You're going to get a worksheet on dihybrid crosses. And here's another one you can do for practice if you're not so sure. And if you're still struggling, then shoot me an email and we can look at it. But these are two pretty good examples for how you do it. I think the trickiest thing may be figuring out how we get these combination of genes going in for the sets of parents outside the Punnett squares. So remember, above and down the left, that's where the parents' genes are. The 16 rectangles here, those are the possible gene pairings in the offspring. Doesn't tell us we're going to get 16 offspring produced. It tells us the odds for certain genotypes when an individual cross happens. All right, good luck with this.